Students, in the last class we discussed the last part of the biotechnology. Now this is the last class. In this class we are going to discuss the remaining part of biotechnology, including biomedical technology. Now in this class we are going to discuss the cancer biology, the stem cells and biomedical technology. And this part also is a miscellaneous topic what we discussed in the previous classes. From these topics we are getting one question, particularly from the biomedical technology we are getting one question. Now as usual, while discussing the questions, around each and every question we have to discuss some important points. Now as we observe the first question, now the assertion is the cancer cell shows a tendency to become spherical shape. The reason: cancer cells exhibit metastasis. Again I am repeating the assertion. The cancer cell shows a tendency to become spherical shape. Now how our body is going to get the cancer or tumor? In our body a continuous cell cycle is going on. The cell cycle included, one we call it as G1 stage, S stage, G2 and M. In order to regulate the cell cycle, different genes are present. One we call it as CDC2, CDC13, CDC28 and P53 genes. In mammals, the 4 kinds of genes are present. Other than mammals, only 3 kinds of genes are present. Whenever the cell cycle control is going to last regulation, the cell continuously divides. If the cell continuously divides, that leads to formation of a new group of cells. That we call it as neoplasm or tumor. Now here one assertion is the cancer cell shows a tendency to become spherical shape. Now as we observe the cancer cell, that appears to be spherical in shape. What is the reason for this assertion? The less number of microfilaments. If microfilaments are less in number, the cancer cell appears to be spherical in shape. But here what is the reason? Cancer cells exhibit metastasis. So A and R are correct, but R is not the correct explanation of A. Now as we observe the cancers or the tumors, these are of the 2 kinds. One we call it as malignant tumor and the second one we call it as benign tumor. We are discussing mainly the malignant tumors. In the malignant tumors, around the tumor there is no capsule. But in case of benign tumor, around the cell group a capsule is present. So in the benign tumor the cells do not migrate from one place to another place. The benign tumors can be removed by surgery, whereas malignant tumors cannot removed by surgery. In the malignant tumors, around the mass of the cell there is no capsule. So from one place to another place the cells migrate. That we call it as metastasis. So malignant tumors exhibit metastasis, that is correct. Why the cancer cell or tumor cell is spherical in shape? Due to less number of microfilaments. So based on this information, the cancer cell shows a tendency to become the spherical shape, that is assertion correct. the malignant tumor cells exhibit metastasis. So A and R are correct, but R not the correct explanation of A. Now the next question. An example of liquid tumor. The tumors are broadly classified into: one is carcinoma, the second one is sarcoma, the third one is leukemia, the fourth one we call it as lymphoma. Generally, the cancer is going to occur in our body to what type of cells? Epithelial cells. Because in our body the epithelial cells divide rapidly. Generally, the cancer or tumor is going to attack those cells divide rapidly. Particularly in our body, the epithelial cells. If the tumors are associated with epithelial cells, they are named as carcinoma. 85% of the tumors are carcinoma. What about the sarcosoma? Those are derived from mesoderm. For example, bone derived from mesoderm, cartilage derived from mesoderm, blood vessels derived from mesoderm. If the cancer is going to attack mesodermal derivatives, they are said to be sarcosoma. What about the third category? Leukemia. The leukemia is popularly known as liquid leukemia. Now here an example of liquid tumor. One we call it as glioblastoma, that is an example of carcinoma. Adenocarcinoma, an example of carcinoma. Chondrosarcoma is an example of sarcoma. Myelocytic leukemia, that is an example of leukemia. They are popularly known as the liquid tumors. Now the answer is, now we discuss the next question. Cancer of nervous tissue. As we observe the development of organisms, in the epiblast, one type of cells are called the ectodermal cells. We learned in the previous classes. Some of the ectodermal cells will become the ectoderm, that is one type of germ layer. What about the remaining ectodermal cells? The remaining ectodermal cells will become a plate called the neural plate. The neural plate develops the neural folds. The neural folds unite to form a neural tube. The anterior part of neural tube becomes the brain, posterior part becomes spinal cord. So this entire part we call it as the nervous system. So the nervous tissue is derived from ectoderm. So here cancer of nervous tissue is a kind of, here the nervous tissue is derived from ectoderm. The ectodermal cells are going to get the cancer. They are said to be a type of carcinoma. One we call it as sarcoma, that is going to occur in the parts derived from the mesoderm, and carcinoma associated with epidermal or ectodermal cells. Now the nervous tissue is derived from ectodermal cells. So the cancer of nervous tissue is a kind of carcinoma. Now the answer is, the next question. A malignant tumor of secondary lymphoid organs. We discussed in the previous classes the immunology. What are the cells of immunology? One we call it as B cell, the second one T cell and NK cell, and mononuclear phagocytes, polynuclear phagocytes. All these are discussed in the previous classes. What are the primary lymphoid organs? One we call it as bone marrow, the second one is thymus with reference to human being body. Whereas in case of the birds, what is an example of primary lymphoid organ? That is bursa fabrici. Here we are asking, a malignant tumor of secondary lymphoid organs. What are the secondary lymphoid organs? One we call it as spleen, the second one we call it as lymph nodes, and the third one we call it as MALT, mucosa associated lymphoid tissue. So these three are an example of secondary lymphoid organs. If tumor is going to occur in the secondary lymphoid organs, that we call it as lymphoma. What is an example for the lymphoma? One example is Burkitt lymphoma. Now here, a malignant tumor of secondary lymphoid organs. If the tumor is going to occur in the secondary lymphoid organs, that type of tumor we call it as lymphoma. Here the example for the lymphoma is the Burkitt lymphoma is an example of the tumor going to occur in the secondary lymphoid organs. Now the next one. The next question, assertion. Cancer cells show a tendency to become spherical. Why the cancer cell is become spherical? In the cancer cell, the microfilaments are less in number, so that the cancer cell appears to be spherical. So for this assertion, the reason is due to less number of microfilaments, so that the cancer cell is spherical in shape. Here we observe the reason. Cancer cells have abnormal antigens on the cell surface and unusual number of chromosomes. So the reason and assertion both are correct, but reason is not the correct explanation of A. The next one. Neutrophil is formed from. Now as we observe the stem cells, this is hemopoietic stem cell. From the hemopoietic stem cell we are getting myeloid progenitor and lymphoid progenitor. This is hemopoietic stem cell. It is going to give myeloid and lymphoid progenitors. Now this myeloid progenitor gives rise to committed progenitors. One we call it as granulocyte monocyte progenitor, and the second one we call it as the basophil progenitor, and the third one we call it as the platelets progenitor, and the next one we call it as erythroid progenitor. The hemopoietic stem cell gives rise to myeloid progenitor and lymphoid progenitor. This myeloid progenitor gives rise to committed progenitors. One we call it as granulocyte monocyte progenitor, and the second one we call it as basophil progenitor, and the third one we call it as thromboplast progenitor, or the material regarding the thromboplasts or platelets progenitor, and the fourth one we call it as erythroid progenitor, and the fifth one we call it as the neutrophil progenitor. As we observe the lymphoid progenitor, the lymphoid progenitor gives rise to one we call it as B cell, and the second one we call it as T cell. Now here the neutrophil is formed from the hemopoietic stem cell gives rise to myeloid progenitor. This myeloid progenitor gives rise to 5 kinds of committed progenitors. Of 5 or out of 5 committed progenitors, one type we call it as neutrophil committed progenitor. From this neutrophil committed progenitor, a functional neutrophil arises. In the neutrophil, the nucleus is many-lobed. It is called microscopic polysemen. In the WBC, the neutrophil number is highest. These are said to be phagocytes. They perform phagocytosis and provide protection to our body. So, the functional neutrophil derived from the neutrophil committed progenitor, that in turn derived from the myeloid progenitor. Here the answer is, the neutrophil is formed from myeloid progenitor, or it is also said to be myeloblast committed progenitor. Now the next question. Which of the following are not pluripotent cells? In the stem cells, what we learned? One we call it as the zygote. In case of animals, the zygote is a totipotent cell. What is meant by totipotent cell? The zygote has to give entire organism. Now from the zygote, the pluripotent cells, the multipotent cells, unipotent cells will arise. What are the pluripotent cells? They give almost all types but not all kinds of cells. Again repeating, the pluripotent cells gives almost all cells but not all kinds of cells. What about the multipotent cells? They give few kinds of cells. What is meant by unipotent cells? Unipotent cell gives always one kind of cell. Here we are asking, which of the following are not the pluripotent cell? Embryonic stem cells, the zygote, the hemopoietic stem cells, and these three are said to be an example of pluripotent. They are going to give almost all but not all kinds of the cells. Now here, out of these four, which is not an example of the pluripotent? The B cell progenitor. It is not a pluripotent. The B cell, it is derived from the lymphoid progenitor. Later the B cell divides into two cells. One we call it as plasma cell and memory cell. So the answer is, the B cell progenitor is not an example of pluripotent. Now the next one. Arrange the following in a right sequence of form a functional cell ultimately. Now the first one, granulocyte monocyte progenitor, myeloid stem cell, neutrophil, embryonic stem cell, and hemopoietic stem cell. Now as we observe the myeloid stem cell, that is nothing but the myeloid progenitor. The myeloid progenitor is going to give the embryonic stem cell, or the embryonic stem cell gives rise to hemopoietic stem cell. The hemopoietic stem cell gives rise to myeloid progenitor. The myeloid progenitor gives rise to granulocyte monocyte progenitor and neutrophil. Now you have to observe the order. Now regarding this question, as we observe the first one is the embryonic stem cell. It gives rise to the hemopoietic stem cell. The hemopoietic stem cell gives rise to myeloid progenitor. The myeloid progenitor gives rise to granulocyte monocyte committed progenitor, and it gives rise to neutrophil committed progenitor. the granulocyte monocyte committed progenitor, it gives rise to functional monocyte. The monocyte enters into tissues and become macrophage. What about the neutrophil committed progenitor? It gives rise to the functional neutrophil. So embryonic stem cell gives rise to hematopoietic stem cell, myeloid progenitor, granulocyte monocyte committed progenitor, gives rise to monocyte. The monocyte enters into the tissues, become the macrophage. Neutrophil committed progenitor gives rise to the functional neutrophil. That is one type of development of the cells. Now the answer is the next one. Arrange the correct sequence of erythroid lineage. Now this one we call it as the hematopoietic stem cell. The hematopoietic stem cell gives rise to this one we call it as myeloid progenitor. The myeloid progenitor gives rise to 5 kinds of committed progenitors. One we call it as erythroid progenitor. This erythroid progenitor gives rise to proerythroblast. The proerythroblast gives rise to erythroblast. Now it gives rise to reticulocyte. It gives rise to functional erythrocyte, that is RBC. Now as we observe the production of the functional RBC, it is also called erythrocyte. The hematopoietic stem cell gives rise to myeloid progenitor. The myeloid progenitor gives rise to 5 kinds of committed progenitors. One among them, erythroid progenitor. It gives rise to proerythroblast. It gives rise to erythroblast. It gives rise to reticulocyte. Finally, we are getting the functional erythrocyte. Now, as we observe the development of RBC, the development of RBC takes place in one line. If the evolution takes place in one line, that is technically called the lineage. What is meant by lineage? The evolution takes place in one line. Now the answer is. Now we go for the next one. Stem cells present in the smooth cells. Now as we observe our body, in our body 3 kinds of muscles are present. What are the bulk muscles? They are said to be voluntary muscles. In our body, majority of muscles are voluntary muscles. The voluntary muscles are also called striped or striated or skeletal muscles. Now, as we observe the voluntary muscles, in the voluntary muscles, one cell we call it as satellite cell. Now, the second type of muscles we call it as smooth muscles. What are these smooth muscles? Their function is not under our control. The smooth muscles are also called visceral muscles or involuntary muscles. Now in the smooth muscles, what is the regenerative cell? The pericyte. Whenever the voluntary muscle cell damaged, the satellite cell gives rise to voluntary muscle cell. Whenever the smooth muscle cell is damaged, the pericyte gives rise to smooth muscle cell. Now, the satellite cell always gives rise to voluntary muscle cell, whereas the pericyte always gives rise to the smooth muscle cell. So, these two are an example of unipotent cells. what we discussed just before the previous question. Pluripotent, they give almost all but not all kinds of cells, and multipotent, they give few kinds of cells. What about the unipotent? They always give only one kind of cell. The best example is satellite and parasite. Now here the question, the stem cells present in the smooth muscles are, the answer is the parasite. The next one, assertion, zygote is totipotent cell. In case of animals, as we observe the zygote is said to be totipotent cell. In case of animal's body, the totipotency having only for the zygotic cell. In case of plants, the meristematic cells are said to be the totipotent cells. And other cells also said to be totipotent cells in case of plants. Take for example sponges. In case of sponges, what are the totipotent cells? The archaeocytes. In case of earthworm, what are the totipotent cells? Basal cells. In case of all well-developed animals, the totipotency power has only to the zygote. The zygote at the power of totipotency, it undergoes cleavages, become morula, blastula or blastocyst, becomes gastrula, embryo, fetus, young one and adult. So the zygote is a totipotent cell. It can give rise to the whole organism. So both are correct. RE explains A. Now the next one, the progenitors that are formed in bone marrow and differentiated elsewhere. Now as we observe the formation of functional B cell and functional T cell. Now in our body, this part we call it as the bone marrow. This one we call it as the bone marrow. This is an example of primary lymphoid organ. The primary indicated by this symbol. Now in the bone marrow, what is present? The pre-B cell. In the bone marrow itself, the pre-B cell becomes the mature B cell. M means mature. Now the mature B cell enters into secondary lymphoid organs. What are the examples for the secondary lymphoid organ? We already learned the spleen, and the second one is the lymph nodes, and the third one we call it as MALT, mucosa associated lymphoid tissue. Now the mature B cell enters into secondary lymphoid organs and become the functional B cell. Now go for the T cell. Now this one we call it as the bone marrow. It is an example of primary lymphoid organ, we know very well. In the bone marrow, first of all, the pre-T cell develops. Now this pre-T cell enters into the thymus. This is a type of endocrine gland. In the thymus, the pre-T cell becomes the mature T cell. Now the mature T cell enters into secondary lymphoid organs. There it becomes the functional T cell. Later the functional T cell divides into TH and TC. Later the TC will become cytotoxic T lymphocyte. What about the B cell? It divides into the plasma cell and memory cell. Okay. Now as we observe the B cell, the pre-B cell becomes mature B cell in the bone marrow itself. What about the T cell? The pre-T cell develops in the bone marrow, where it becomes mature T cell in the thymus, but not in the bone marrow. So you observe the question. The progenitors that are formed in the bone marrow and undergo differentiation elsewhere, that means somewhere, the answer is the pre-T cell. Pre-T cell develops in the bone marrow. In the bone marrow, progenitors are present. From the progenitor, we are getting the pre-T cell. The pre-T cell enters into the thymus, a type of endocrine gland. In the thymus, it becomes the mature T cell. But whereas the B cell, the pre-B cell becomes mature B cell in the bone marrow itself. Now, the next one. Arrange the following in sequence during the formation of RBC. The question is repeated. Now, this question is discussed in the last one. As we observe the hematopoietic stem cell, it gives rise to myeloid progenitor. The myeloid progenitor gives rise to erythroid progenitor. The erythroid progenitor gives rise to proerythroblast. That gives rise to erythroblast. That gives rise to reticulocyte. That gives rise to the functional RBC or erythrocyte. Now based on this, you observe the answer. Now the answer is the fourth one. What is the fourth option? First, myeloid progenitor. That gives rise to erythroid progenitor. That gives rise to proerythroblast. That gives rise to erythroblast. That gives rise to reticulocyte. Finally, we are getting the functional RBC or functional erythrocyte. Now the next one. Which of the following cells can directly arise from lymphoid progenitor? What are the cells directly arise from the lymphoid progenitor? Now this one we call it as hematopoietic stem cell. The hematopoietic stem cell gives rise to myeloid progenitor, lymphoid progenitor. The lymphoid progenitor gives rise to committed progenitors. Here what are the committed progenitors? One we call it as the B cell. Second one we call it as T cell. Now the T cell, what we discussed in the later. First of all, the pre-T cell. Later it becomes the mature T cell. Later it becomes the functional T cell. Later it divides into TH and TC. Finally, it becomes cytotoxic T lymphocyte. What about the B cell? In the bone marrow, first of all, we are getting the pre-B cell. It becomes the mature B cell. Later, it becomes functional B cell. Later, it divides into plasma cell and memory cell. Now, here we are asking, what type of cells directly arise from the lymphoid progenitor? One we call it as the dendritic cell. And the second one we call it as NK cell. What is meant by NK cell? That is natural killer cell. Now you observe the answer. Which of the following cells can directly arise from the lymphoid progenitor? The answer is dendritic cell and NK cell. NK means natural killer cell. Whereas we observe the myeloid progenitor. From the myeloid progenitor, we are getting what type of committed progenitors? One we call it as granulocyte, monocyte committed progenitor. The second one is basophil progenitor. The next one is neutrophil progenitor. And the next one we call it as the thromboplast progenitor and erythroid progenitor. From the myeloid progenitor, what will arise directly? The dendritic cell. Here we should not explain the natural killer cell. Now the next one. One among the following is not a descendant of myeloid progenitor. That means one among them is not derived from the myeloid progenitor. One is the natural killer cell, second dendritic cell, third basophil, and the fourth neutrophil. You observe this table. This is myeloid progenitor. From this granulocyte monocyte progenitor, it gives rise to functional monocyte. Later it becomes functional macrophage. Basophil progenitor, later it becomes functional basophil. Neutrophil progenitor, later it becomes functional neutrophil. What about the thromboplast progenitor? Later it becomes the functional thrombocyte or functional blood platelets. Erythroid progenitor, finally it becomes the functional RBC or erythrocyte. Now, from the myeloid progenitor, what is directly arised? The dendritic cell, but not natural killer cell. Here you observe the lymphoid progenitor. From the lymphoid progenitor, what kind of committed progenitors arised? B cell, T cell. And what will arise directly? Dendritic cell, natural killer cell. Here we should not explain the formation of natural killer cell. So the answer is the next one. Most human tissue and organ transplants are. Now we are discussing the transplantation. Now you observe the options given in the question first of all. Most human tissue and organ transplantation. One is allograft, xenograft, isograft and autograft. Now as we observe, what is meant by autograft? For example, in the body of one organism, here the skin is damaged. Now this damaged skin is replaced by another skin from somewhere in the body. That means here the skin is damaged due to burning or because the acid is poured. Once acid falls on our body, here the skin is damaged. This damaged skin replaced by another skin from our body. That means in the same person body the transplantation takes place. That we call it as autograft. What is meant by isograft? Genetically identical. Transplantation between individuals genetically identical. Generally it is going to occur between twins, kavala pillalu. And the allograft, transplantation between genetically not identical but the same species. And what about the xenograft? Xenograft means transplantation between two different species. One species genetically not identical, allograft. Genetically identical, isograft, generally going to take place in the twins. What about the autograft? One person, the transplantation takes place. Now here we are asking, the most human tissue and organ transplants are. The answer is allograft. That is a transplantation between non-identical of the same species.